the University of Pennsylvania, we have a fairly comprehensive molecular imaging program that involves a variety of modalities that include MR, optical imaging. We're starting to explore molecular ultrasound probes. But one of the things we're trying to do is really broaden the multidisciplinary approach to molecular imaging. And so I would say one of the things that characterizes the Penn program is really a collaborative cross-disciplinary approach. That said, you'll hear almost entirely about PET today.
Well, FDG has proved to be a great radiotracer, very versatile, but in this new age of precision medicine, there's a lot of therapies that are directed. And to select patients for these targeted directed therapies, there's other radiotracers that we could use to say that this potential therapy may or may not work. So for patient selection, we could use different radiotracers, select patients that may benefit from a specific treatment, and then use those selectively in those patients, as well as measuring the effects of treatments early on. So if the treatment is not going to work, we won't continue. So it can change treatment rather quickly. So these targeted methods, I think, is more of precision medicine, personalized medicine.
So biomarkers in breast cancer and in other cancers are incredibly important. We have a huge number of treatments available now, and trying to figure out who should get which treatment is the big question.
So really, I see it as two big problems. One is we're giving women treatments that perhaps they don't need, perhaps over-treating. And on the other hand, there's a group of women where our treatments are not effective, and we may not find that out until the end of chemotherapy if we've given them neoadjuvant therapy and at the time of surgery. Or we might find that out years later when the cancer recurs. And I think functional imaging can help us with both of these problems today.
And we're really now beginning to focus quite a bit on the balance between proliferation and quiescence using Bob's tracer, F18 ISO1, which we have now a couple of trials ongoing with in addition to preclinical work. So overall, not only can we measure glucose metabolism and find tumor sites, but the program here and around the country demonstrates the ability for us to be able to broadly characterize tumors, gain insights in the lab that we wouldn't otherwise gain, and in particular take these new methods into the clinic with the goal of more individualized, more precision approaches to the treatment of cancer patients, and in our case, with a focus on breast cancer.
I currently work on multiple projects within our training program, from imaging proliferation with the sigma-2 receptor to actually using the sigma-2 receptor as a mechanism of delivering alpha-emitting radionuclides.
Our interest in the sigma-2 receptor was really around coming up with a way for imaging what we call the proliferative status, which is the ratio of cancer cells that are actively dividing or proliferating to cells that are driven into quiescence by nutrient deprivation. Now, histologically, that measure is done by looking at a protein called Ki67.
So this is where functional imaging comes in. If we could image this marker, Ki67, which is a reflection of cellular proliferation, then we could look at expression in the whole tumor. We're not bound by sampling error. And also in the case of metastatic disease, we could look at intertumoral heterogeneity, that is the difference between tumors. So imaging gives us a whole picture of the patient.
After we made this initial identification of the sigma-2 receptor having this potential to study this concept I call proliferative status, we went about doing two things. Number one, we had to validate that, and we had to do a series of studies to show that the density of this receptor really did reflect cell proliferation and that there was a lower density in quiescent tumor cells. And we published these studies over the past 20 years. We have 3 or 4 publications demonstrating this. And the second thing we need to do was identify a ligand that could bind to the receptors. And that proved to be a challenge initially because there were no lead compounds for us to develop PET probes on. So it took us a while, but we eventually identified some compounds that had very high affinity and high selectivity for sigma-2 receptor versus other receptors. And that compound is called F18 ISO1, And that's the probe that we're currently using here at Penn. It's in a breast cancer trial. We plan on expanding it into other indications, and it's also being used at Washington University in imaging studies and in clinical trials.
This aspect of functional imaging helps us in the case of undertreatment. We believe that the expression level of that marker as measured by ISO1 will be predictive of response, and especially to some of the newer drugs like cell cycle inhibitors that are on the market. And so we have some very exciting preliminary results, and that is how functional molecular imaging will help in breast cancer treatment.
So I think our biggest challenge is going to be as a community to making sure that we pool our resources and use them in a rational way to make sure that we get individual tracers from that initial idea all the way through clinical use, rather than having hundreds of things that never make it past those initial stages. And so the biggest challenge there is in terms of trying to decide what to move forward, because we are resource limited, of course, and trying to figure out how to best use those resources is the challenge.
And finally, through the help of Komen, through the help of the institution, through the help of our department, we're able to support the development of careers of young investigators, young scientists, many of whom you'll hear about in other interviews today, that again are the lifeblood of our field. And right now, in an era of challenging funding, it's of the utmost importance for these folks to get support and mentorship to be able to keep the specialty going, and that's a very important focus of what we work on here.
I think Penn is unique primarily because we have an amazing mechanism to go from basic science all the way to the clinic in a very short amount of time, and being able to see that whole spectrum, I think, is an experience that I don't know if it happens in very many places. So I'm very grateful for being able to actually view the whole spectrum of the translational science process.
From the preclinical development to the validation phase, so preclinical into a human, into clinical phase. And it's a very collaborative environment. Everybody's very supportive. And when we encounter certain problems, we can also both sit down and try to work out the problems and make sure everything would go smoothly before we actually would launch certain things. So that collaborative environment, I think, is really crucial in what we're doing.
As a clinician currently practicing right now in nuclear medicine, I think it's very important to understand the background science of what's going on, to really appreciate the radio tracer, understand its limitations, and understand really the biology that's going on. But also important is, as a clinician, to talk to the basic scientists and be in this translational realm where we could see what's important for patients, where there is a clinical need, so that something could be developed. So I think the back and forth going both ways is very important, and the future of medicine, where I would like to be.
And the thing that really I enjoy most about this field is that it's really at the cutting edge of medicine and biomedical research. That I feel the probes that we are developing and have developed can be used to study important biomedical questions. And when we take a compound and it makes that transition from a structure on the whiteboard in my office to doing the synthesis in my lab to doing the preclinical evaluation in animal models, and then finding something that works every step along the way, and then doing a phase 0 clinical trial in patients, and to see that it works in patients, you really can't describe the feeling one gets when you see that. It's really an unbelievable feeling. I think that's the feeling that drives me every day, and I think that's the feeling that people could get and chemists could get if they came into this field.